Welcome to Genomics Gupshup. Genomics Gupshup is an initiative by MapMyGenome to create a community around genomics and to simplify genomics for everyone. And we do this by bringing together experts from allied areas like medicine, genetic counseling, research, nutrition, fitness, and more. Please join us as we spread the word about this exciting science of genomics. And we are now available on your favorite podcast as well. So just search for Genomics Gupshup on Apple, Spotify, Amazon, or Google Podcast. So today we are on the 45th episode of Genomics Gupshup, and I'm so excited that we have somebody very special today, Dr. Sendurai Mani. He is an Indian-American oncologist, and he's a molecular biologist who's currently the Associate Director for Translational Oncology at the Legorreta Cancer Center at Brown University, and also a Professor of Pathology of Lab Medicine at Warren Albert Medical School at Brown University. He is known for his research on cancer stem cells and metastasis, and he was the first to demonstrate that cancer can make more cancer stem cells by reactivating an embryonic program known as epithelial to mesenchymal transition, or EMT. We'll talk a little bit more in the course of this discussion. And his research has also led to the development of new therapeutic strategies for targeting cancer stem cells and preventing metastasis. Dr. Mani is a highly respected scientist. He has a postdoc from MIT and was at the MD Anderson Cancer Research Center for several years. He's also received numerous awards for his work, including the V Scholar Award from the Jimmy V Foundation for Cancer Research, the American Cancer Society Research, and the MD Anderson Research Trust Award. He's also a fellow of the American Association for the Advancement of Science. He's passionate about making precision medicine available to everyone by combining clinical, pathological, and genomic disciplines to bringing personalized medicine to cancer patients. Dr. Mani, a seasoned researcher, is also committed to investigating ways to target cancer stem cells responsible for developing metastasis and chemoresistance. I have known Dr. Sendurai Mani since MIT days and then also at MD Anderson Cancer Center. So, Dr. Mani, it's an absolute honor to have you on Genomics Gupshup. Welcome to the show.
Thank you. No, thank you for having me. I really appreciate the initiative, what you are all doing. This is a great initiative to educate or bring awareness to the public. I thank you for what you're doing. Genomics, yes, yeah.
So, Dr. Mani, you have done a lot of work on many, many different things. But I want to understand when Dr. Mani was a little boy, what got you excited into this whole molecular biology and cancer and all of that? What inspired you to get into this journey of science?
Thank you. I really appreciate this question. I am always curious. That's one thing which about me is that I'm curious to know how things work. And so when I was an undergrad, I took an exam where I got the first mark in the class, even though I wrote the exam half in Tamil and half in English. Wow. Because it was an English college. It's a college. Everything was in English. But I wrote half in Tamil and half in English. The professor did not score me for English. Rather, he scored me for my knowledge. So that's when I realized that I can understand well. English is a language which we can learn. But understanding a concept it's a gift. So I was able to understand things better. So I realized this is probably the passion or road in which I should go. And then that's what I've been doing.
That's great. So all our t-shirts say curiosity is in my DNA. So I guess it is in your DNA. And then what happened? You were doing your undergrad, and then you did your PhD, and then you went to the US. And so can you tell us a little bit more about how that transition happened?
Sure, thank you. So I completed my undergraduate at a college in Madurai, which belongs to Madurai Kamaraj University. So when I joined, I wasn't sure whether I would be able to survive because, everyone was speaking an amazing English and this and that. But then when I joined, I realized that I could understand things better. So then towards the end of my undergraduation, I wanted to pursue master's education. So I applied for a master's program to various schools, but I was fortunate to get into an integrated biology at Madurai Kamaraj University, which is one of the very unique program where you kind of you get a lot of practical knowledge rather than theoretical knowledge. So there I met with the two amazing individuals. And one person name is Venkat. He kind of taught us how to think differently to solve a science problem rather than thinking traditionally using a memory-based system. And which kind of fits well with my kind of an idea of analytical rather than a memory-based system. Then I met another person with the name Pradeep Kumar Chaliao, who became my very good friend, and he also became my mentor. So he helped me to explain things quite a lot and then devoted a lot of his time to make sure that, I go to the next level. So that's when I learned that there is a school called Indian Institute of Science, which is the #1 institute in the country in India for research and education. So I applied to that program for a PhD. There are 2,000 students all around the country apply, and they select only 20 to 25 out of that. And again, I was lucky that I managed to get a PhD program into the Indian Institute of Science. And I also was lucky to get a PhD under somebody with the name Professor G. Padmanabhan, who was the director of Indian Institute of Science, and some greatest, simplest human being and a great scientist. And in his lab, I worked for a PhD for 3-4 years or 5 years. When I was doing a PhD, there was another professor also in the lab who was also co-mentored me. The name is Dr. Rangarajan. So when I was finishing my PhD, this was mostly on a biochemistry and a molecular biology, my PhD was. When I was finishing my PhD, they invited Robert Weinberg to give a talk at Indian Institute of Science. And at that time, I went to the professor, Robert Weinberg. He was the first one to ever clone the first oncogene, the RAS oncogene, the first tumor suppressor gene, and the enzyme called human telomerase was cloned in his lab. And he was the first one ever to make a cancer cell in a lab by genetic manipulation. So he came to give a talk in India. I approached him. I asked him, can I come to his lab? Even before that, I showed him the work, what I have done for my PhD. And he said he was going to talk to me. And at that time, there was another professor came and said that, can I steal Bob for a few minutes? I said, okay. So Bob said, Mani, I'm sorry, I need to go. I will be back soon. So they were supposed to come back in 30 minutes or an hour, but they came back after a few hours. I was waiting in the same place, and Bob Weinberg recognized that I don't give up. So that's all was my interview was, that he gave me an opportunity to go to his lab at MIT. So when I joined Bob's lab, I did not know much about cancer biology. I knew molecular biology. So I wasn't sure whether I will be able to survive in that lab. But luckily, over a period of time, I published a few papers with him. And those papers ranks or has the highest citation among all the paper Bob Weinberg ever published in his life. So I'm very happy and proud. So from there, after spending quite some time in his lab as a postdoc, I moved to MD Anderson Cancer Center as an assistant professor. And I spent 15 years there, got promoted from assistant to associate to full professor. Then I recently, 9 months ago, I moved to Brown University.
That's a fascinating journey. And I wanted to also tell some of our viewers that, I've known Dr. Mani for many years, but I didn't realize how prolific an author he was and how much impact he had created. So for those of you who don't know all these scholar profiles, Google Scholar profiles, and their impact that he has. He has what they call as an exceptional, H index and the I-10 index. So for those of you who are interested in research, you definitely must check out. And maybe, we'll start off with, you talked a lot about things that people might not be very fully aware of in terms of the telomerase and the paper that you were talking about, which maybe it will be great to maybe have a little discussion around what that was about and why was it so impactful in the course of, just understanding cancer as a science.
Thank you. So it was in 1998, 99, I started my postdoc work at the Weinberg Lab at MIT. So right around the time, they were transforming human cells, normal human cells, into cancer cells. So even before that, people did transform normal cells to cancer cells, but when they did, they add a chemical to a normal cell, and then you convert a normal cell into a cancer cell. So when that happens, you have no clue what had happened inside the cell for that cell to become a cancerous. So, but that's not going to help us to treat the patient, because we need to know exactly what you need to make a cancer cell. So there was a postdoc at the time, he's a professor now at Dana-Farber, his name is Bill Hahn. So he took those cells, he introduced 3 set of genes. One is called SV40 early region, another one's called RAS oncogene, and the telomerase. So why he did that? So the SV40 oncogene, or SV40 oncogene, which inactivates 2 tumor suppressors in the cell. It's called RB and P53. And then he puts an oncogene, which is a HRAS, and then he puts a telomerase. So why do we need to do all these 3? In order for a normal cell to become a cancerous, so there are multiple roadblocks they have to cross. One of them is, this is called a tumor suppressor. They have been stopping the cell going crazy. So you need to inactivate it so that the cell can become a tumor cells. But even if they get a tumorigenic ability, but they still need to proliferate. So that's where one need to give this something called oncogene or mutation in an oncogene, such as HRAS. But even then, the cell has a limited lifespan, because there's something called a telomeres, which keeps a clock, just like the way we live for a particular number of time. The human cells also has a particular number of days they can live, or number of times they can divide. At that point, they have to die. And they figured out that you need a telomerase to add a telomere. They got a Nobel Prize as well for that discovery. So then you add a telomerase, then you can make a normal cell into a cancer cell. So perfect. So that's when I joined the Weinberg Lab. And most of the study, another thing I want to bring to the audience, your audience, is that there are 3 major types of cancers. There's a carcinoma, it's a sarcoma, and it's the liquid tumors. So we mainly focused on carcinoma, which account for more than 80% of all cancers, which includes breast, prostate, pancreatic, lung, liver, spleen, and whatnot. So in these tumor, when you put a SV40 early region, telomerase, and oncogenic HRAS, you convert a normal cell into cancer cell. But those cancer cells, when you put them, let's say breast cancer cells, into the mouse breast, they form a tumor in a mouse breast, but they don't go anywhere. But cancer patients don't die because of the breast cancer alone. They die if they have a metastasis to a different organ. So that's when I got curious why these cells form a tumor locally, but they don't go anywhere. So I started researching, working with another colleague at that time, my name is Jing Yang. We figured out that in order for these cells to go from one place to another, they need to break something called cell-cell adhesion, because most of the epithelial cells are they are attached with one another through various cell-cell adhesion molecules. When these cells become a cancerous, that transformation does not break the cell-cell adhesion. Rather, they're still attached. That's why when somebody develops a tumor, they tend to be benign or local. And at some point later in the course, they become migratory, invasive, and metastatic. So the way the tumor cell achieve that by reactivating an embryonic program called EMT. So I'll just to summarize what is EMT. So it's called epithelial to mesenchymal transition. So upon fertilization, the first cell which forms is called epiblast. These epiblasts are epithelial cells. They eventually undergo this EMT or epithelial to mesenchymal program and generate something called the mesoderm. And the same layer also creates an ectoderm and endoderm without an EMT. So the mesoderm gives rise to heart, blood, kidney, ovary, and various organs in the human body. So this play a very, very, very vital role during embryo development. It's off once the embryo is formed, and mostly get turned on during wound healing or during fibrosis. Smartly, the cancer cells reactivate this program to make cancer cells migrate from one place to another. So that's the way we figured out that EMT program play a vital role in making a tumor metastatic from one place to another.
So that's what has happened when you're saying that one, your cancer cells which were, let's say, originally in the breast or somewhere, they're there, but because of the EMT, they managed to move across to other organs, and that's where that whole process is called as metastasis.
Correct. In addition, one other thing. So one other thing which, when let's say a pathologist looking at a tumor in a lung, they can figure it out right away. Just by looking at a tumor in a lung, they can say whether that tumor belongs to the lung, or they have come from breast or prostate or pancreas, just by looking at something called histopathology. In other words, they create an organ similar to where they come from. So that I hypothesized that the cells not only become mesenchymal so that they can gain migratory capacity and also become stem cells. So this allows them to recreate an organ histopathologically similar to where they come from. So that is what one of the major contribution, which the one you were talking about, made a huge impact in the cancer field.
That's fascinating. I think you had also mentioned, I remember in your TED talk about how do you consider a cancer, the whole thing as a car. Maybe you can explain that a little bit more to the audience, because I found that really, it was very succinct and very easy to understand. You're talking about oncogenes and tumor suppressor genes, as well as the telomerase or the fuel. And maybe it'll be great to understand it from you, why you consider that. And also, I think now that you are talking about this whole migration thing and understanding it, I was thinking about it from, maybe when you're looking at it, let's say, I'm from Rajasthan. I now live in Hyderabad, right? So in some ways migrated somewhere. You can still look at your genes and figure out, ultimately where you're from. Like you said, the histopathologist finds out. Maybe is there some way we can sort of simplify your analogy a little bit further, and that will be great.
Thank you. So thank you again. So with respect to the car analogy, so every car the car do have a brake, and you have an accelerator, and there's a gas tank. This is three are the major ingredients for car to go, stop, or stop permanently when you run out of gas. It's very similar. So the tumor suppressor is almost like a brake, where it's constantly telling the car when you go fast, you just apply a brake, then you slow down, or when you want to go fast, you apply press on a gas pedal and you go faster. And that's an oncogene. So imagine a car which is going full speed on a normal speed, and you have a gas tank, and the gas is the car is using it, keep moving. So when the gas tank runs out, again, the car will stop. But at some point, imagine the brake is broken, which is a tumor suppressor, which is no longer functional. And your gas pedal is nothing happened. So then still the car will stop at some point, because even though you don't have a brake, you're going to keep running for some time and going to stop.
Because the fuel is over.
Oh. Fuel is going to run out. But imagine the brake is broken. Well, there's somehow, for some mechanism, the gas pedal is continuously on. So then you're going to run very fast. but still you're going to stop at some point because you're going to run out of gas. Now think about a third scenario where you don't have a brake. You're continuously pressing an accelerator, but most importantly, your gas tank has an ability to refill itself on a constant basis. So at that point, the car is not going to stop. It's going to keep going and going and going and going. That's what happens in a cancer, where you need to do all three. So this is where, many times people ask me, I have a mutation in my tumor suppressor gene, like a BRCA1. Will I get a cancer? So I tell them, you are highly prone to develop a cancer, but you will not develop a cancer just because you have a mutation, because that makes them prone, but not for sure. So that's how a normal cell become a cancer, is because you need to inactivate tumor suppressor gene, activate an oncogene, and constantly fill the gas tank with the telomerase. So
so you're saying that this whole, if you're filling it up constantly, what if it was an electric vehicle? I'm just taking you completely off track now.
A solar panel on the roof,
Yeah.
and then you are, as long as you have a sunlight, you keep going, right? And then keep going, and then so that's what the cancer does. It kind of activates its own machinery. Just one enzyme, telomerase, activate, and most of these genes. I remember you talking about epigenome in one of your previous episodes. Most of the genes are kept silent through epigenetic mechanism. Now, you overcome that by epigenetically modulating the expression of telomerase. So now it's continuously on, the telomerase, which adds a telomere. The telomere is the clock gene, which decides to shorten the telomere. So every time when a cell divides into two daughter cells, the telomere length shrinks. So that goes shrink, shrink, shrink. And at some point, the cell senses, I have a very short telomeres. I should not survive anymore. The cell commits suicide. It's called apoptosis. Now, if there is a way you can add telomeres to the end, and the cell is not even going to know that they have a shorter telomeres, or the cell is already gone crazy. And on top of that, the cell also lost the tumor suppressor genes, in this case, RB and P53, for example, and they are the one who sense whether there is a shorter telomere or not, so that the cell need to die. So the sensing mechanism is gone, and the clock is continuously on. So that's the way the normal cell become a cancers. And then you also asked about how the migration, right? Can we do a genomics to figure out the migration pattern? Beautifully, it's a very nice, amazing question. I'll tell you why. Because until now, a pathologist look at a tumor in a lung, and then by them understanding whether that tumor has created an organ similar to breast or prostate, they make a call whether this is a tumor from breast or prostate. Now we have a new set of pathologists, a molecular pathologist. So who not only look at a histology of the organ, but do the genomic test. So by doing a genomic test, you can figure out at high accuracy whether that given tumor has come from a breast or prostate or whatever the organ. In addition, through genomic approach, which you guys are doing, you could also identify vulnerabilities. So what is a vulnerability? So now we have a number of drugs for these oncogenes. The tumor cells get addicted to the oncogene. And through genomic approach, you can figure out, are these tumor cells addicted to that oncogene, this oncogene? We have hundreds of oncogenes, and we have a drug for many of them. And if you know exactly which oncogene this cell is addicted to, then you give a drug for that. So then you deprive them from that addiction, and they come out, they die, basically.
So essentially, you want cancer cells to die. And in order to do that, you are either looking at figuring out how to stop that addiction that the oncogene might be providing to the thing, or you want to maybe reduce that fuel that you mentioned, or maybe somehow get back the tumor suppressor back up, right? So these are the three options that we have. So which one, in your opinion, is the most fascinating for you in terms of the biggest chances of converting, like you mentioned, you talked about from normal cell to cancer cell, but from cancer cell to normal cell, is that, or rather to kill the cancer cell, which is the most interesting approach from your point of view?
So, reactivating a tumor suppressor gene is very hard, and people are trying that through various genetic engineering approach, where you can reintroduce a normal copy of a tumor suppressor gene, then you can put a break. That's a genetic editing approach people are taking, which is a tougher route, because if you have 1 million cells, you need to do the editing in every cell, right? Now, telomerase is another approach a number of people are doing, which is again great, but there are some drugs which are coming up. But the easy route, which seems to work, is the addiction, right? If you deprive the addiction, they die. But what happens in this process is that when you deprive them of, let's say you have 1 million cells in a small tumor, and then you deprived on many of them, let's say quite like a 0.9 million cells, let's say, are addicted to this particular oncogene, you kill them off, the small percentage of them figure out a way to activate a different oncogene. So while you kill the cells which are addicted, there's a new clone evolves during the process. That's where the current therapy targeting the oncogene works very well, remarkably well. But relapse is the most common problem among these patients, because it gets turned on invariably, but they get to live longer compared to the traditional chemotherapy.
My dad used to tell this, that there was this whole mythological creature, right, where you kill something, but that again comes back up, right? Like, I'm trying to remember which mythological creature, but I think it was somewhere with Durga, or I don't remember, but those little Kali, I think, where you had these things that kept coming back up, right? So in some ways, I think we are still learning a lot about how these are happening, and hopefully we'll get to a point where some of these newer techniques, like that they are coming up, hopefully they'll become a lot more understood, but that won't happen unless we learn or understand the science behind it, right?
Very well said. The most important thing is, for example, olden days, people were scared to give more than one therapy to a patient. It used to be monotherapy. Now we have come to 2, 3, and then people are now talking about cocktail. So when the tumor has a 5 mutation, you can target all 5 mutation.
And from a research point of view, are you also seeing a lot happening? You mentioned that each cell you want to understand more. So are you seeing a lot more happening on looking at individual cells and learning what is going on in each of those? Because clearly, I think there is a lot that is happening on the single cell, and understanding that. A few years ago, I think you had to still see it under a microscope. You had to do, the laser capture and a few other things. But now I think we are seeing more molecular ways of doing it, right? I think you are probably the one who probably knows a lot about these areas of research that are going on. But I know we might be digging into too deep, but maybe it will be helpful to just get a little sense of what is happening in that space.
Thank you. So many years ago, what we did, we took a tumor, we ground the tumor. Just after you grinding it, you figured out what is different in the tumor compared to normal tissue. It gave us some information. But then it didn't tell us exactly, let's say you have 1% of the tumors are the bad tumor cells, and the remaining 99% is the daughter cells. They're not bad, but they're just a daughter cells. But that 1% was buried inside the 99%. But we were able to get some idea at that time with the global sequencing. So then we went to do something called single cell sequencing, where if you have 1 million cells, you sequence all the 1 million cells, or at least representative clones, and you figure out that these cells probably this tumor is made of 10 different kind of tumor cells, and the immune cells and all the cells which support the tumor. Then you go and target them differently. So now we are into something called spatial transcriptomics, where we not only get a single cell information, but spatially imagine a tumor cell sitting, and then there's an immune cell sitting next to it. We know and immune cells are supposed to kill the tumor cells. So that's true when a tumor starts. But over a period of time, the tumor cell convinces immune cells that I'm your friend, don't kill me. So now immune cell, even though sitting next to it, it doesn't kill the tumor cells. In addition, the tumor cells makes the immune cells undergo something called exhaustion. In other words, make them tired. So they not only cannot kill the tumor cells, they are tired most of the time because the tumor cell sets up. Now, by doing the spatial transcriptomics, we not only trying to understand whether the immune cell is sitting next to the tumor cell. How is the immune cells? Is it exhausted? If it isn't exhausted, can we make them sensitive or maybe reactivate them so that now they can go and kill the tumor cells?
Sounds great. It's fascinating how this whole thing, we can now sort of get a nice inner view into what is happening, in cancer. Now,
crazy.
Now, before we get on to understanding a little bit more over there, I just wanted to maybe, it'll be a good time for those people who don't understand, because the difference between what is hereditary genetics, like what you inherit, versus what happens in a cancer, in the case of cancer, where, things might change over a period of time. I think just maybe it'll be helpful, sometimes when we are looking at it from understanding the risk, like you were talking about, we are mostly looking at it from what you inherited from your parents. But in the case of cancer, there is a lot that is happening within those specific tumor cells that is going on. And I think that's why it's so important to be able to understand what is really happening, how is it transforming, and each part is different. And therefore, they say that you're not just having one cancer, you are cancering. And like you mentioned, there's so much happening in this space. So I think, maybe you could explain to us what is, for each of these different cancers, is it genetically, is it, is there a different thing that is happening, or maybe, we can save that for a later time?
Just maybe say it in 1-2 words. There's a sporadic mutation, there's a hereditary mutation. So hereditary mutation contributes to around 5% of all tumors, right? Somebody has a mutation in a gene called APC or BRCA1. So they tend to develop tumor through their lifestyle. In other words, if normal person and then a person with the mutation lives in a particular environment, and both get exposed to similar chemicals, similar food, similar lifestyle, then the person with the mutation will tend to develop tumor faster than the person without a mutation. But just because they have a mutation, it's not that they're going to develop a tumor. It's extremely important to study the genomic profile from like in a company like yours. So that would allow somebody to know whether they are prone to develop a tumor or not. So based on that, one could adjust their lifestyle.
So I think another thing I wanted to maybe talk a little bit about was about cancer incidence. Right? We are clearly living longer now compared to maybe even 10, 15, 20, 30 years ago. But that also means that, given that cancer typically happens at a later age, we are seeing also increase of cancer incidence. But is it a matter of only that because we are living longer, or is the change in terms of incidence a factor of the fact that we understand that there is more cancer going on, we are able to detect cancer, or maybe is it something else, right? Do you see a change in terms of, what is going on in the world? I think you are in the US, but also maybe if you have a understanding of what is happening in India, because, when I look at the statistics, they used to say 1 in 10 and then 1 in 9. Now they're saying 1 in 8 is likely to have cancer. So why is it that suddenly we are seeing this increased incidence of cancer when there is so much that is also happening on the on the molecular side and understanding and the diagnostic side as well? Maybe you can.
Thank you. You articulated all the reason. So as you rightly said, that we do live longer. So therefore, so the probability of cell gaining a mutation over a period of time, you increase the probability, because if someone lives for 40 years, their cell would have divided X number of time. But if somebody lives 8 years, that double the number of time the cell divides or kind of get exposed to various insult over a period of time. So living longer definitely adds more cancer incidence. And then on top of that, the lifestyle, right? We all have, for example, in India, when I was growing up, I did not even know much about allergy, but now allergy is very common. So our lifestyle and then eating all kinds of junk food, I would say, and each one of them going to insult our genome. Mm. So when you are insulting the genome with inflammation, so there are two kinds of inflammation: acute inflammation, chronic inflammation. Acute inflammation is good for us, but chronic inflammation is not. If somebody smokes, they are chronically creating an inflammation in their lungs. Somebody drinks alcohol, they are chronically creating inflammation in their liver. And so things, if somebody is eating like a very spicy food chronically, they are creating chronic inflammation in their stomach. So the chronic inflammation kind of causes the second problem. So lifestyle, right? That matters. And the third, the most important thing is, in olden days, somebody want to detect whether they have breast cancer, they have to develop a breast cancer for them to know whether they have breast cancer or not. But now you go and get a mammogram, and even before it can be kind of detected by any other means, the mammogram detects breast cancer. Now you have a genomic testing. You can detect by looking at a mutation or circulating tumor DNA whether somebody has a tumor or not. So adding all these 4 factors, we are able to add more number. But one should not worry because of this number. One should feel happy that I have a proper testing approach these days. So I should get tested early so that breast cancer and prostate cancer in the US, if it's detected early, the success of survival is almost 99%. So one need to get tested early rather than late.
Absolutely. And I think that's what, we keep trying to tell people, that information is not going to make your cancer come or go. Information can make you more aware, and therefore you take the right kind of, you go to your doctor faster. And I think a doctor can do a lot more for you when you go there early versus when you go there at stage 4, when by the time is when your metastasis has already happened, right? And then it's harder. So I think that's something that most people think that, if they don't know, then ignorance is bliss, but it's not necessarily the case. You mentioned that 99% have a positive this thing once they go to a doctor early with breast cancer. I think similarly, I think for other cancers also, we are seeing a lot more better survivals when they come to see a doctor early. And you also mentioned a lot about lifestyle-related risks that are there, and people don't necessarily believe. They always think that cancer means if it's in your genes you will get it, but that's not true. And I think I read a study which was a fairly large study which said that almost 50% of those cancers may be preventable, right? So do you believe that that's true, or you think that people can, if they were more aware, they could prevent more cancers?
Yes, so that's what I've been promoting. Awareness is the key. Awareness is the key, and then taking an action, right? And somebody says, yes, drinking alcohol can create more inflammation in my liver. I may develop a liver cancer. But, being aware and doing a precautionary measure versus I know I'm going to get potentially may get a liver cancer, but I'm still going to drink alcohol, right? So it's an attitude comes at the end. Those who care, they take the preventive measure. Those who don't care, they continue with what they want to do. And then they cry later at some point when things happen and, oh my God, I should have stopped earlier.
Yeah, I think that's something that, hopefully awareness will increase as we go along. So one of the things that, for instance, we do a genetic predisposition, we help people understand that. But I remember the first time, it was a friend of mine who got this test and said, they got a risk for lung cancer, right? Now, the first reaction at that point was, he was coughing, it must be lung cancer. And I explained that is not, coughing can be due to many other things, but it was not about scaring you that, this is something that is there, but it was more about saying that if you look at the kind of things that you do, whether it is smoking or other things, that's a lot of risk that you can take out from your possible diagnosis, right? So when someone finds out what would be, from a genetic test that they are at risk, let's say they're only 25, 30 years of age right now, and they find that they have a risk for, let's say, and a curable cancer, like a breast cancer, or let's say something that has lower survival rate. What do you think should be the, what would be the next steps for those families or somebody who's just found out that they have a risk for some potential cancer?
So again, they have a mutation which kind of predispose them to a cancer, but that doesn't make them cancer, right? They don't allow, it's not that they're going to develop cancer. So the minute they come to know, it's a good idea to take a precautionary and preventive measure. So Angelina Jolie went and, for example, she did a mastectomy. As soon as she came to know that she has a mutation in her BRCA1 gene. And one need not go to that extreme, but one could do, for example, at that point, think about include exercise in their lifestyle, include a healthy diet in their lifestyle, include happiness in their lifestyle, because most of the time stress causes a lot of additional mutation as well. So one need to reduce the stress. So those are all will be beneficial when they come to know this. But if they don't know, they are going to keep doing what they do. They are going to literally a car running without a brake. Mm-hmm. That's what the mutation in this tumor suppressor gene is nothing but. So they still have an option for a gas tank to run out. But why? If you can figure out a way to, go on a right road and figure out you apply less gas rather than more gas, then you still go slow. Rather than, you apply full gas, well, there is no brake. Mm-hmm. So that's what your company is doing, telling the people that, your brake is gone. So be careful. You apply gas a little bit, be careful, live long, healthy life rather than developing a tumor in a short span.
Absolutely. I think you put it so beautifully, so easily. So hopefully, I think you should definitely, understand this whole car analogy and how this, how you can actually make those changes in your lifestyle. And I think those changes can ultimately result in your car going in the right direction, in the right place at the right time, I guess.
Yeah.
So maybe we can move to the microbiome. I think you have done some research in microbiome as well, right? Can you tell us a little bit about, what is the connection between microbiome and cancer, if there is any?
So the microbiome is one of the most important element in our human body. I think we have something like 36 trillion cells, and 36 or 38 trillion bacteria or microbiome, not bacteria, microbiome in our body. So they all work together in a very symbiotic way. So when you go and disrupt the symbiosis, you do create problem. For example, olden days, people would think twice before giving an antibiotic. Now, for anything and everything, they just give an antibiotic. And you just wipe out the microbiome, right? And then the microbiome need to be reconstituted. In olden days, we all used to take milk or yogurt, not milk, the yogurt. Now, we drink or take yogurt which are sterilized, which is a yogurt which is made into yogurt through microbiome, but they kill the microbiome so that they don't continuously grow and spoil the yogurt. So you get a yogurt which has no microbiome. So you are not reconstituting that microbiome, which is good for your body. So this is where there was a beautiful talk last week I listened to by somebody with the name Gordon Freeman, who discovered something called PD-1 and PD-L1, which is a very important drug target now for immunotherapy. So he gave a talk. He's my good friend and a collaborator. So he was saying that you give a patient an antibiotic, then give an immunotherapy. And to another patient, you don't give an antibiotic, give an immunotherapy. Guess where it works better? The patient who did not receive an antibiotic. So the microbiome do help the immune cells to kill the tumor cells. If you ask me how, we are trying to understand it. But it's clear, the data says microbiome is a very important player in various aspects of our human life. So all those who are listening, I would strongly recommend that reconstitute your microbiome if you ever have to take an antibiotic. And when you reconstitute, use a yogurt. That's plenty, but not a sterile yogurt which you buy in a store. A homemade yogurt would be perfect.
I was just going to say that maybe you recommend that we make our yogurt at home and eat it. That's how we have always done. I remember even when I was living in the US, I remember that, one of our aunts, we went to her house, got the yogurt and made sure that we were making our own yogurt. Of course, occasionally you can, I used to buy store-made yogurt, but I think on an average, on a regular basis, I think we still eat the homemade yogurt. And
if you buy from a store, there is something called a live and active culture. Yogurt with a live and active culture would be okay. But again, they don't really give you spectrum of microbiome because they add selective species. But yeah, thank you.
Yeah, so I think now we are also trying to get people to understand which probiotic will work for them based on their microbiome, so that they can actually take that probiotics, prebiotics. But I think naturally, if they can include some fermented foods and other things, I think it's always better. A lot is also today, a lot is happening in terms of AI in medicine. So do you think that, especially given that cancer is so complex, do you think that, using AI in cancer will help us in understanding this science better? Or do you think that there is still a lot more we need to do to understand, the actual biology for individual cells before we can really transition into using AI?
So it's a very important question, and thank you for asking that. So it's a parallel universe. So one need to understand the tumor better, because that is going to help us to understand how the tumor has become rogue, right? But human mind cannot comprehend all the information what we are getting. So somebody who's studying a tumor suppressor, when you talk to them about something else, while they may know, they may not know, but it becomes too much information. So, when I was starting my PhD, there were like 20 different journals which were publishing cancer-related stuff. Now, probably 20,000, I don't know. There are too many. I don't even know. I cannot keep track of how many journals out there publishing paper in cancer. So you don't even know. So there is not even a single soul can read all those papers, put them in mind, and understand how things work. So this is where AI will be extremely helpful. So I do have a company. I started a company. It's called Ilon Precision Oncology. So what Ilon does is that, let's say a patient has a mutation in a tumor, and they have a 50 mutation. Not all oncologists would know among those 50 which is the dominant mutation. In other words, which mutation the tumor is more addicted to. So we need to target that. then AI approach will help. But in order for AI to work, you need data. So what the ILAN does, it uses or it takes help of an expert who does this day in, day out. They are academic physicians. They do research in the lab. They treat a patient in a clinical trial. So we take their help. We get information. We give them this 50 mutation, and they look at it and say, okay, this patient may benefit from drug A targeting mutation 10, for example. So we feed that information into AI. So over a period of time, we would have got a lot more information as to if somebody has this kind of mutation, what drug they may benefit from. But today, we have not. So radiation, there are pathology, there are immune cells, so many factors. We need to add them into this AI to come up with a kind of a solution which would help the cancer patient. So both are extremely vital and need to go in parallel.
So before we use AI, we also simultaneously need to use NI, or natural intelligence of experts like yourself, actually, to be able to understand and give that information to the patients or the physicians that are there. I know that there are so many things that I can ask you about, but there is one thing that we find, which is at least in India, that a lot of times most doctors are familiar with like a BRCA mutation because thanks to Angelina Jolie. But if you had to name maybe 4 other mutations, or 4 or 5 that you like or like to research on, rather, not like as in want to have, but what would those mutations be for, if you can take any cancer or any mutation that is there?
So there's a mutation called APC. It's called adenomatous polyposis coli, something like that, APC.
How do you remember these names?
So the APC, patients with APC mutation tend to develop colon cancer. And of course, BRCA1, there's a BRCA2. There's one particular mutation which is really kind of difficult to target, which is very bad, which is called P53. So which is also tumor suppressor. So every one of them are tumor suppressor. The mutation tend to occur in a tumor suppressor in case of hereditary cancer. Okay. So P50, there is a patient had a tumor in one organ, and then they treated that, and then the same patient developed a tumor in a different organ. They treated that, and then they developed a tumor in a different organ. So when the P53 is supposed to be called a guardian of the genome, and so when somebody has a mutation in P53, they tend to develop all kinds of tumors. So it's very important to figure out somebody has a P53 mutation. There's something called retinoblastoma or RB gene. They tend to develop retinoblastoma. So we can go on. The list goes on, but these are the dominant one.
Yeah, I remember. I think P53 is extensively talked about even in Siddharth Mukherjee's book for understanding of cancer and how it was under discovery of the whole molecular mechanisms as well. Okay, so what are your predictions in terms of the future of genomics in cancer care?
So this is like, we say bread and butter, right? This is one of the most important thing which happened is the genomics. So when Eric Lander was sequencing the human genome, when he proposed the idea, everybody kind of laughed, like, it's not possible. But then I remember I was a postdoc at the time at the Whitehead Institute. There was one big building where so many robots are breaking the human genome into small pieces, putting them in a bacteria, sequencing them, and putting them in a supercomputer at that time, and then assembling the data to create a gene signature or the genome. But now we can do that, like, you can send the DNA and you guys probably can sequence the genome for 1,000 people in one day, right? We are in a different era. So it's keep evolving. And the price also dropped from $100 million to probably $1,000. Maybe it's going to go down even further. So So genomics is really, it's like a cell phone of cancer care, right? So olden days, communication was a big deal. But now everyone has a phone. It's almost like that. The genomic information, very soon, people will understand easily the genomic information. My mom, who never went to school, she's talking about genes. So that's the way things are going to go. The information is going to flow. I think both has to go in parallel. Genomics is it's a central to, in my opinion, to our understanding of cancer going forward.
And I also, moving to a completely separate topic, I also saw one talk that you had done, which was about social determinants of health and how they play a role in cancer. Can you tell us what these social determinants of health are? I know you've sort of mentioned a few of them over the course of this discussion, but it would be helpful for people to understand that, you mentioned happiness and a few other things. Maybe it would be helpful to understand what all of these social determinants are.
Thank you. So social determinants is like, where do you live, for example. So Rhode Island is one of the first industrialized state in the US. So they dumped a lot of waste, a lot of pollution all around the state. Now, when you look at the cancer incidence in Rhode Island, where the state where I live, some county where the incidence is like a #1 in the country. And if you look around, and there were industries before there in that area, right? So same way, when somebody is working at a cotton mill and they are constantly inhaling pollution, so where do we live makes a whole lot of difference. And the pollution is one of the major driver. And the family where you live, whom you are living with, what do you eat? So your genome is going to contribute so much. And where you live, all these various factors is what is going to add more complexity to it. So the social determinants of health is nothing but understanding your social environment and all the factors which can contribute. Somebody has a BRCA1 and they live in a worst social determinants of health environment, they are going to make it a tumor faster than somebody who lives in a better SOTH, which is social, SDOH, social determinants of health or environment. So many factors. One thing I could even say, for example, in olden days, we all had A2 milk, which is the milk which is supposed to be less inflammatory. But now, so the A1 milk, the cow which produces A1 milk, they produce 5 times more than the cow which produces A2 milk. So everybody switched for the economic purpose to an A1 milk. So you get only A1 milk when you go to a store. But A1 milk is more inflammatory. That triggers all kinds of problems. So the farmer or whoever, the person who's making money is able to make more money, but that person also drinking that A1 milk and having all health-related problems. So one has to draw a line. Do I want to make more money or do I want to have a good life? So it's a complex social dynamics.
I absolutely. I think, when you look at, I used to often write that your health outcome is determinant on environment, your lifestyle, and your genetics. So I think, your social determinants, a lot of it is the environment, not just in terms of physical environment, but also your social environment that you live in, because culturally there might be some norms that maybe you're following. That could be one. I think you mentioned about Rhode Island being an industrial area, but even in India, I think there is a particular train I remember that is called the cancer train, because from a particular location, there was part of it was the environment. And I think, that would come to even my hometown called Bikaner, and that was called as the cancer train, right? So it comes to Punjab and a few other areas. And part of it was primarily environmental. It was not that people in those areas had higher genetic risk, but it was also environment that caused that. So maybe we'll move on to the next round of this. I know we are almost at the hour, so I wanted to take it to the next. Unless you want to maybe add something before, no? Okay. So this is something I ask every guest. In your opinion, what is prevention? Or how would you prevent something?
So it's very important to have a knowledge, right? You cannot prevent unless otherwise you have a knowledge. So knowledge is a fundamental to everything. So for example, am I prone to develop a cancer? One. So for example, doing a genomic testing will help to get that information. So once you know you are not or you are in, so once that information is cleared, ask a question that why I am doing what I'm doing, rather than blindly following. Somebody said that, even if it is a doctor. So I went to a doctor. This is just a small incident. I went to a doctor, and then the doctor said, I need vitamin D. And I asked the doctor, do you know? And the doctor asking me to take a very high dose of vitamin D, very high dose. I said, why? He said, that's what is recommended. I asked the doctor, do you know whether vitamin D is water soluble or fat soluble? The doctor said, I don't know, and didn't even know. So if it is a water soluble, if you take any dose, it'll get excreted through the urine. If it's fat soluble, it has to be converted into water soluble by liver, and then it has to get excreted. So it's very important to kind of pay attention to the details. If it's a fat soluble, I would prefer to take what is necessary for the day rather than big dose, because you're getting loaded with so much of vitamin D until it gets excreted. So things like that. So it's very important to kind of pay attention to the details, and then through that one could lead a normal, healthy life. So for prevention, understand the science behind it. Need not be in a you read a science paper, but using a simple common knowledge, one could get that.
Absolutely. I agree. I think, there are a lot of papers that talk about toxicity because of high doses of vitamin D and other such cases that we see. Now, the next question is, what is your secret to healthy aging? Because you look the same as I remember 20 years ago. So what are your secrets to healthy aging?
Thank you. My wife is very much into nutrition. She does quite a lot of research on, balanced diet, the microbiome. So I would give a lot of credit to my wife. So plus, control calorie and what kind of calorie, right? Whether you're getting a calorie from an ice cream or you're getting a calorie from a brown rice. It's two different things. It's very important to watch out the calorie, how much one consumes. The Western diet is very addictive. So we try our best to avoid, try to eat healthy. So that's the secret, plus exercise. And then, not to get stressed too much about what goes on around everywhere. So try our best to kind of take some time off and do the right thing.
Absolutely.
Live like a normal human being.
Yes. Again. So I think, it's very hard, especially when you go to like an American grocery store, and now actually even in India, because the choices that you're getting are so many in terms of processed food and in terms of different kinds of things that might have a lot of stuff hidden into there. So it's kudos to be able to understand and then be able to implement those. So what is digital health? Is there a thing called digital health? And what do you think that is?
So I think that's the next wave. Digital health is kind of you're basically adding an AI component to your health. So the one of the thing which I learned recently is something called a Gemini, where they're trying to create a digital of you, or replica of you. So in a digital health, you are kind of having number one, AI built your own lot of information put together and create information about you. And then somebody understanding that information, helping you. So in a digital health, you have a combination of all this, getting help and knowing who you are.
That's great way of putting that. So I'm sure you've heard a lot of myths, right? Whether it's in molecular, whether it's in cancer or anywhere else. Are there any fun ones that you remember that you've heard people talk about in terms of myths that are propagated as being true in medical or healthcare?
Can you just reframe that?
So what are some of the myths that you like, which are not true, but people constantly say? For instance, I think a lot of times the doctors talk about, how, especially gynecologists, you talked about how what you're supposed to eat, what you're supposed to do during childbirth could be some example of myths. In cancer, there might be myths, right? Like people think that this is true, but it is not necessarily true. So
thank you. So at some point, nobody believed that immunotherapy will work. Mm-hmm. Nobody, immunology was like, kept as something which, never thought that you can cure melanoma today, right? Because some time ago, I remember many people saying they focused on a tumor cell, but not on immune cells, because they felt immune cells has nothing to do with the tumor. There are very few who believed it, but many did not. But today, that discovery not only got a Nobel Prize, but it's changing, transforming the lives of so many people, and people become cancer-free, not getting a tumor relapse later. They're just becoming cancer-free. So that's one of the things which I have seen. We thought that it's not going to work, or it's not even playing a role in this process. Now it's transforming the cancer care.
Oh, absolutely. I think it has totally changed. And maybe, what are the kinds of immunotherapy, if maybe that you can tell us? I know this was not supposed to be this thing about immunotherapy, but it's a brand new area where I think a lot more, and it's, I think, more happening in the US rather than in India. So you mentioned that, the immune cells are basically coming to your rescue in some form. Maybe if you can tell us a little bit more about that.
So in a tumor, so let's say normal cell becoming a cancerous, as soon as that happens, the immune cells come to that site to stop the tumor cells. So it does, they do. But at some point, the tumor grows bigger and bigger, and then it switches the microenvironment. It's called immune microenvironment, from tumor suppressive to immune suppressive. So there are a lot of immune cells which are not tumor killing, they become a tumor promoting. So how this happens, right? So it's kind of a chronic inflammation which happens. The tumor is considered as a chronic inflammatory wound. That's the way some people claim, which is true. So when you have a chronic inflammation, the immune cell's job is to come and kill anything bad. On a normal day, when the killing is over, you need to come and stop the immune cells, say, now you are done your job, it's time to stop. So there is a mechanism in a normal day. But in a cancer, that it's a time that you need to stop, that signal is lost. Sorry, the other way around. When the time comes, you need to stop. Instead, that signal is permanently turned on. So therefore, the immune cells comes there, but it forgets it's need to kill the tumor cell because it's got a signal from the tumor cell saying, you don't have to kill, the job is done. So that's called a PD-1, PD-L1, or many such communication between tumor cells and immune cells. We have many such communication. So what immunotherapy does is that communication has been broken by something called immunotherapy or antibody-based therapy. So you break the communication. Now the tumor is no longer telling the immune cells that the job is done. Rather, the job is still need to be done. So now the immune cell kill the tumor cells. So you break that communication. That's all one is doing. So there's also CAR T cell, where again the T cell is going and killing a tumor cell. But for that, the CAR T cell technology, you could say, imagine you want to kill people with, let's say, black color hair. I'm just giving that as an example. So you have a T cells for the black color hair, and then it'll go and kill everyone who has a black color hair. So same way in a tumor, you need to have a specific antigen. It's called antigen, a small signal in any tumor cell which has that signal, and this T cell can recognize that signal and kill that tumor cells. So that's a CAR T cell. So there are also using a various immune cells. You can use the cell-based therapy. So this is various type of immune therapy, which right now in practice, but the most common one is breaking the communication. It's called immune checkpoint blockade therapy.
That's great. I think you explained everything so in nice and simple terms that I think that's kind of like, it is always nice to ask you all these complex things to explain those. And I think that's the whole purpose of Genomic Gupshup. So thank you for explaining all these complex topics in very, very simple terms. So I'll now move to the rapid fire. And I don't think you know any of the questions I'm going to ask. I'm going to ask you different ones now. So if you had to describe cancer in one word, what would that be?
It's an enemy can be fought.
Okay. That's a good one. What is the most common complaint about you by your friends?
I work too much.
Okay. Are you a morning or an evening person? I think I know the answer, but
I'm an evening person, but I came morning for you.
thank you. What hobbies do you have other than molecular biology, cancer, and anything to do with the lab?
So I travel a lot. I like to travel around the globe. For example, last month I was in Germany driving on an autobahn, going like as fast as I could. And then, went to a mountain, just went on a hiking in one of those Swiss Alps. So, I like to be in outdoor when I find time.
That's great. So was that your favorite place to go to, or do you have a special place that you like to travel?
I like to travel various parts of the world and learn. I've been to China many times. I've been to various parts of the world, and then you go and see and learn how people live and what do they eat. I used to think that pizza or the way we make pizza in the US is a little different than the pizza made in Italy. Only when you go there and eat the pizza in Italy, you realize that how different it is. And so we like to live the local life, live there for some time, be part of the community, learn from them.
That's great. I think it's always nice to learn from different places and continue to grow. So one last question. How do you continue to be so curious all the time?
I don't know, maybe it's in the genes. I like to find how things work, and I enjoy that. That's what is keeping me going. In morning you wake up, constantly that goes on in the background, like how, why, and how, and it helps.
Absolutely great. So thank you so much. I think it has been a really great episode. We've had lots of discussions on some very complex things, but you've explained them in very simple terms. So thank you. I think cancer is not an easy topic for most people to understand. So it is very nice when it can be broken down at the cellular level, but you've also created a lot of nice analogies for people to understand. thank you for all the great work that you have done. I think that has also created a lot more over the last 20 years. You mentioned that paper was done sometime, early, almost 15, 20 years ago. So I think since then, all the great work you have done, thank you for doing all of that. And hopefully we'll see a lot more happening from your lab that is coming in, and as well from all the companies that you've created. So thank you, and thank you for being on Genomics Kaksha. Thank you.
I just want to thank you. This is a great initiative and a great idea. It's a great service what you're doing. And in addition to providing the genomic sequencing through your MapMyGenome, this is one of the very, very key contribution. I thank you for what you're doing. Thank you.
Okay.